Thank you. It's a real honor, of course, to be here, to be able to introduce Shinya Yamanaka at this Pan-Pacific meeting, which has been fantastic. Several weeks ago, when I was asked if I would be willing to introduce Shinya, I, of course, said yes. One doesn't get the opportunity to introduce a speaker of such stature very often. But as time approached, I started to wonder, of course, what am I going to say that hasn't been said 1,000 times before? And panic started to set in. So, it's really up to date. It's been, interesting to kind of think about how to approach this kind of introduction. Obviously, you don't want me flipping through his CV and telling you where he did his postdoc. So I started to lose a lot of sleep. But then I decided to take a trip to the future, because I figured that would be a good way I could get a perspective on how Professor Yamanaka's research is viewed by posterity, perhaps giving me a little bit of an insight into his work that might have not been shared by other people who have shared this kind of difficult task of introducing him. So I landed up in the end of the 21st century, and I was wandering around, and of course I was there surrounded by robots of high intelligence, and of course all artificial intelligence is running everything at the end of the 21st century. But at the time, of course, they call it intelligence, not artificial intelligence, because human intelligence is, sort of quaint and outdated. But I ended up speaking with my friend, the robot Dave, who I really wanted to say, so what is known about Shinya Yamanaka at this time? And how is his research viewed? And surprisingly, he said to me, well, not many people actually know who Shinya Yamanaka is. And I said, that can't be possible. He said, yeah, well, he actually suffered the fate that of his own success and his own recognition and being famous that ended up leading to his obscurity. And I said, well, how can that be? And I didn't really understand it until he shared this slide with me, this image. And of course, you'll recognize these as all major brands that became so popular and so famous and so well recognized that people stopped using them by their names and basically referring to all things in that category that shared those characteristics. So things like you'll recognize Q-tips and chapstick and jacuzzi, and you might be surprised that like dumpster and laundromat and even dry ice, who knew? So these became generics, and in fact, Dave said that's exactly what happened to Shinya. So basically, in the future, when people refer to Yamanaka, unfortunately, they're not referring to you, Shinya, but to something generic that really is whatever a Yamanaka might be. And of course, I was very curious to try and figure out what the meaning of Yamanaka might be at this time. And that's when Dave introduced me to the Future Dictionary. And there in the Future Dictionary is Yamanaka with a small y, basically an entry into this dictionary. And of course, we're all waiting to find out, well, what does it mean? And there were actually many entries, not just one, and each with a quote, contemporary quote of how Yamanaka was used at the time. So the first entry was a noun. So a Yamanaka is a discovery so profound that it revolutionizes a field. And the accompanying quote that was in the common parlance was, discovering how to induce human hibernation was the Yamanaka that allowed this man-Jupiter mission. So it wasn't just a noun, though. It also turned out that Yamanaka is a verb, to Yamanaka, and that is to pursue with unmitigated passion. And the associated quote with that verb, and for the basic aficionados in grammar, it's the participle form of the verb, but the PhD student mapped every neuron in the human brain, Yamanaka-ing her way to insert a claim. Okay, but it didn't stop there. Yamanaka also turns out to be an adjective, which is characterized by intense focus and dedication. And the quote is, the runner's training regimen was so Yamanaka that he finished his next marathon in 2 hours. And for those of you who are not marathoners, 2 hours is sort of what the 4-minute mile was at Roger Bannister's time. So there's one more entry, and you'll have noticed that all of these quotes end in exclamation points, attesting really to the impressiveness of all of these, what a Yamanaka is, or to Yamanaka, or the essence of being Yamanaka. And so that final entry is an interjection, and basically an expression of surprise and delight. And the associated quote with a footnote that basically Yamanaka comes to replace Eureka in the lexicon is, Yamanaka, I've discovered the secret to quantum entanglement. So I think, all of these definitions are not a surprise to anybody in the audience, who everybody is familiar with Professor Yamanaka's work. I mean, Dr. Yamanaka is the brilliant mind behind the discovery of induced pluripotent stem cells that not only earned him the 2012 Nobel Prize in Physiology and Medicine, but really gave hopes and continues to give hope to millions of people around the world for the advances it leads to in terms of regenerative medicine. And Dr. Yamanaka's career is really emboldened by the notion that discoveries are not only about great ideas, but they really are about dedication, persistence, curiosity, and the willingness to challenge the status quo. These are features that Dr. Yamanaka has manifested throughout his career. So please join me in welcoming Professor Shinya Yamanaka.
Okay. Wow, thank you very much, Tom. That was the most sophisticated introduction I've ever had. Thank you. I've known Tom for a long time. I respect you, not only as a scientist, but also as a runner. Actually, I'm not sure you remember it or not, but like 10 years ago, you taught me a very nice training course in San Francisco. And since then, whenever I'm in San Francisco, I run that course. It's like, how many miles? 30K, so 16, 18 miles. So it's a very nice course. And because of that, I'm doing very good at marathon. Actually, earlier this year, in February in Japan, I broke my own record. I ran Beppu Oita Marathon with 3 hours, 19 minutes. So I'm still making a progress. So my running time is decreasing a bit by bit. So according to this curve, I should be able to break 2 hours in 2060. I will be 98. So I have to be live very long. That's why we have been working on iPS cells. So I work at both places in Kyoto, of course, and also in San Francisco. In Kyoto, I work for Center for iPS Research and Application, CiRA. In addition to this, we started a new public interest incorporated foundation, public foundation, CiRA Foundation, 5 years ago, which I will describe a little bit later in my talk. And also I have a lab in San Francisco, Gladstone Institute of Cardiovascular Diseases. My activity in San Francisco is regarded as an on-site Kyoto laboratory. So San Francisco and Kyoto are well connected, just like UCLA and also UC San Diego. So as Tom mentioned, 19 years ago, we reported this new stem cell technology, iPS cells. All we need is a combination of these 4 genes. By introducing these 4 genes into skin or blood cells, we can convert somatic cells back into the embryonic state. They become almost like ES cells, embryonic stem cells. They are nearly indistinguishable. Only one difference is the origin. We don't have to use embryos to generate iPS cells. Instead, we can generate iPS cells from each of you. So 2006, we published mouse paper, and 2007, we published human paper. So once they become iPS cells, they have these 2 important properties. First, we can expand iPS cells as much as we want. And then we can differentiate iPS cells into many types of cells that exist in a body, like brain cells, heart cells, liver cells, pancreatic cells, and so on. So because of these two properties, these are very useful in medicine and also biomedical researches. Like today, I have many opportunities to talk about iPS cells, but actually I did not make iPS cells by myself. It was many young members of my lab who generated iPS cells. So we, once again, we published human iPS cells in 2007. 3 years later, Kyoto University decided to start a new research activity, new research center, which is Center for iPS Cell Research and Application in 2010, 15 years ago. We have approximately 700 people in these 3 buildings in Kyoto. We have one common vision, purpose. It is to bring iPS cells to patients. There are 2 major medical applications of this technology shown here. So once again, we can make iPS cells from patients or from healthy donors. We can expand iPS cells as much as we want, and we can generate a huge amount of brain cells, heart cells, muscle cells, and so on. So by transplanting these brain cells into patients suffering from brain disease, we should be able to recover the function of the patient brain. This approach is called regenerative medicine. The second equally important application is drug development. Instead of transplanting back into patients, we can use these neurons, heart cells, muscle cells from patients suffering from brain, heart, or skeletal muscle. Or we can keep culturing those cells in laboratories of universities or pharmaceutical companies. We should be able to recapitulate diseases. And then we should be able to search for small chemicals to stop the onset or to stop the progress of diseases. That second approach is called drug development. So let me talk a bit about each of these 2 medical applications. So let me start with regenerative medicine. 19 years ago, when we published iPS cells, the Japanese government started to give us huge support. So I specifically asked the government to make a kind of all Japan team so that many Japanese scientists can work as one team, not independently. I believe it is very important. Our main task was to produce GMP-grade iPS cell lines. I was, and I am still a scientist in basic science. So I have never generated GMP-grade materials. So I thought it would be like at least 10 times more difficult to generate GMP or clinical grade iPS cell lines than making research grade iPS cells. To make research grade iPS cells, as many of you know, all we need is one hardworking student or postdoc. And it won't take too much money. But I thought it would be 10 times more, but it turned out to be wrong. We realized it was at least 100 times more difficult to generate GMP-grade iPS cell lines. So in 2000, I believe 2015, we became able to ship the first GMP line to users. We're so happy. So we took this photo. The cells are in this box. So you can appreciate how many people were involved. Nearly 100 people are required to generate just one IPS research grade, clinical grade GMP iPS line. So it is really 100 times more difficult. And it costed a lot, almost $1 million to generate just one line. So I think this is a very good example how difficult it is to move from a bench to the clinic. This is just one example. I was the director of CiRA, and one of my reasons of headache was how to maintain these 100 people. These people do not publish papers. So in universities, it is kind of difficult to appreciate their hard work. In order to give them a better employment, we decided to start a new activity, which is CiRA Foundation. So this is, once again, Public Interest Incorporated Foundation. The name is very similar, CiRA and CiRA Foundation, but CiRA Foundation is started by Kyoto University, but it is now independent from Kyoto University. So those 100 people moved from Kyoto University to CiRA Foundation so that they have a more stable employment at CiRA Foundation. We can pay them a bit better salary. But then I realized I'm now directing CiRA Foundation. So I hope those 100 people are happier. But now I have another headache. Kyoto University will never bankrupt. CiRA Foundation may bankrupt. So I'm not sure if I did the right thing or wrong thing. I hope those 100 people will be happy for a long time. I'll do my best. So anyway, now CiRA Foundation is providing these three kinds of GMP-grade iPS cells. We started with this first strategy. We generated iPS cells from HLA homozygous donors. I don't have time to explain in detail, but HLA homozygous donors, they can cover, just like one donor can cover many recipients. Cover means to provide HLA matched iPS cell lines. So we have generated 4 lines, and just these 4 lines can cover up to 40% of all the Japanese population. That means almost 50 million people, Japanese people, can receive these HLA homozygous iPS cell stock. We are also working on genome-edited iPS cell stock. So we started HLA homozygous donors, and by changing the genome a little bit, we expect that only 10 lines could cover the entire world population. I will explain this a bit later. And now we are working on this also, the third approach, which is my iPS cell project. So this is autologous iPS cell line. We are also working on this. So as for the first approach, we published this paper already 3 years ago in Med. So you can, if you read this paper, you can know the details of this approach. But once again, we can cover up to 40% of all the Japanese population with just 4 lines. But in order to cover the remaining 60% of the Japanese population and also the entire world population, we would need hundreds of lines. So it's not feasible. That's why we started this another approach, genome-edited iPSC stock. So this is there are multiple ways in making genome-edited iPSC lines. This is our method developed by Dr. Akitsu Hotta in CiRA and also Dr. Kaneko. So in this approach, we knock out HLA-A and and B, and by knocking out C2TA co-activator, we can suppress all of class 2 MHC, but we leave HLA-C allele intact. This is important because this retained HLA-C can suppress NK cell-dependent missing self immune rejection. And also this HLA-C can show some antiviral and anti-tumor immunogenicity. So that's why we leave one HLA-C allele. But HLA-C allele is less diverse. So that's why we believe by having 10 HLA-C haplotypes, just 10 is enough to cover more than 99% of the world population. So since 2 years ago, 2023, we started distribution of GMP lines of genome-edited HLA-C retained iPSC lines. And the third approach is my iPSC project. So once again, like 15 years ago, it was very difficult to generate autologous iPSC line. It was very expensive, and it took almost 6 months. But we have been collaborating with many companies. So now we have introduced this kind of automated machine. These are all closed system. So at least theoretically speaking, we can now produce 1,000 iPSC line every year with only 10K, $10,000 per person. So this is where we are about my iPSC projects. So once again, CiRA Foundation is now providing these 3 types of GMP-grade iPS cells. So the vision of CiRA Foundation is to provide the best iPS cells at affordable prices. This is available not only in Japan, but worldwide. So if you are interested in, please check our website. So once again, this first approach, HLA haplotyped iPS cell stock, we have distributed these lines to almost 100 scientists and companies in Japan and also overseas. And actually, they are already being used in clinical trials. More than 10 clinical trials are ongoing in Japan. And most of these clinical trials are using our iPSC stock lines. And in addition to this, many other clinical trials are ongoing in the States, in China, and in Europe. These are just clinical trials in Japan. I don't have time to explain all of these. Let me give you just a few examples. The first one is Parkinson's disease, led by Professor Jun Takahashi, our colleague, who is now who is the current director of CiRA. So from our HLA homozygous iPSC stock, they have generated dopaminergic neurons and they have started clinical trial in which they inject dopaminergic progenitors into the brain of patients suffering from intractable diseases. Just a few weeks ago, they published the outcome of their trials in Nature. According to that, they confirmed safety. They did not observe any major complications such as tumorigenicity or severe immune rejection. And they observed improvement in symptoms 4 out of 6 patients. So they will apply approval to the Japanese government very soon. We have to see how the Japanese government, more precisely PMDA, which is a Japanese equivalent of FDA, will respond. But I think this is very promising. The other example I want to talk about is this one, cancer immunotherapy, which I believe you heard from Dr. Shin Kaneko yesterday. He has multiple projects. This is only one of them, but this is the fastest one. This is already in clinical trial. So he used NK cells. He generates NK cells from our HLA homozygous iPS cell stock. But in iPS cells, he put this CAR antigens recognizing GPC3 tumor antigen. And then they generate NK cells. And they, in this clinical trial, they inject this CAR NK cells into the abdomen of patients suffering from the end stage of ovarian cancer. So I don't know the result yet. I hope Dr. Kaneko will announce the result in the near future, but I believe these approaches are very promising. So let me move on to the second equally important application of drug development. I will give you one example from Kyoto University and another example from Gladstone Institute. So this is one example of drug development in Kyoto University. It's the target disease is ALS, amyotrophic lateral sclerosis. So as you know, this is a tragic disease caused by progressive motor neuron death. So the onset is very minor. Patients feel weakness and numbness in the thumb or some other parts. So in the beginning, it is difficult to have a proper diagnosis. Within a year or two, they start having difficulty in speaking and in swallowing. And then within another 2 or 3 years, they start having a hard time in breathe. So without respirator, they cannot survive. So at the end, they lose all the mobility, whereas they can still think, they can still sense. So it's a really, really tragic condition for the patient and also for the family member. There is no cure, unfortunately. Many pharmaceutical companies have been working on this tragic disease. They have developed many effective drugs on mouse models, but unfortunately, none of them turned out to be effective on human patients. So for this disease, mouse and human are very different. We have to have human model. But each of motor neurons is essential. So we cannot obtain motor neuron from patient because each motor neuron is essential for proper movement. But now we have iPS cells. We don't have to use patient's motor neurons. All we need is a small amount of blood cells from patient or skin cells. From blood or skin cells, we can generate iPS cells. So our colleague, one of the deputy directors of CiRA, Professor Haruhisa Inoue, he has developed a very efficient method to generate motor neurons from iPS cells. So in patients, it takes very long. It takes 30 years, 40 years before they develop symptoms. So in the beginning, when we started collaboration with Dr. Haruhisa Inoue, we thought we may have to culture iPS cell-derived motor neurons for 10 years or for 20 years to recapitulate motor neuron death. But it is not the case. Dr. Inoue and many other scientists found that as soon as they generate motor neurons from patient iPS cells, they start dying. So in the body, I think motor neurons are somehow protected. But in Petri dish, there's no such protection. So as soon as we generate motor neuron, they start dying. So this means we can recapitulate the disease, motor neuron death, in Petri dish. For patient, we can test just one drug at a time. But we can generate thousands of Petri dishes having patient's motor neurons at the same time. So that means we can test thousands of different chemicals at one time. That's exactly what Dr. Inoue did. He first tried existing drugs for other, approved drugs for other diseases to see whether any of them can protect, I can hear something maybe, to protect cell death. And he found one chemical, bostenib, which is a well-known anti-leukemia drug, is effective in preventing motor neuron death. Because bostenib has been widely used in clinics already, it took only, I believe, 2 years to move from this bench-side experiment to clinical trial. Only two years, I believe. So they started phase 1 clinical trial 2019 at Kyoto University Hospital. They talked to PMDA. They decide not to have blind control, but they designed this protocol because each ALS patient is very different. In some patients, the progression is very rapid, but in other patients, it's very slow. And it's sometimes it's not, there is some variation even in one patient. So you have to be very careful in order to see whether the effect you are seeing is really the effect of the drug you are testing. So they selected these 9 patients because these 9 patients prior to the application of bostenib showed a very similar progression of the disease shown here. So this is before the beginning of bostenib. And 3 months after, they started adding bostenib. And they found 5 out of 9 patients, the progression of the disease stopped. Whereas in the remaining 4 patients, it did not stop. So it's not for all patients, but for about 50% of patients, it seemed to be effective. And what was very important in this study was that we may be able to predict which patient is responder and which patient is not responder by measuring neurofilament L level or in their blood in advance. So all of these 5 patients who responded showed lower neurofilament L level in the blood prior to the application of bosutinib, whereas these 4 non-responders showed higher level of neurofilament L. So this may suggest we may be able to predict responders. So based on this result, they started bigger phase 2 clinical trials 3 years ago, and it's still ongoing. So I hope we may be able to provide this new medicine to ALS patients in the very near future. So in addition to this, there are 5 clinical trials ongoing in Japan, as far as I know of, for the drug discovered with this kind of approach using patient-derived iPS cells, including Alzheimer's disease, Pendret syndrome, ALS. Actually, 2, in addition to Dr. Inoue's clinical trials, Dr. Hideyuki Okano of Kyoto University, they are testing another drug for ALS and for FOP and for some cystic disorder in kidney. So as I mentioned, our activity at Gladstone is one of the 3 US on-site laboratory of Kyoto University, Gladstone, UCLA here, and UCSD. Actually, I forgot to mention that Dr. Jun Takahashi's clinical trial has started in San Diego as well, so as a part of this activity. So let me briefly talk about the activity at Gladstone. So at Gladstone, many laboratories are using iPS cells for the second purpose in disease modeling and in drug development. So let me give you just one example by the president of Gladstone Institute, Deepak Srivastava. He is a cardiologist, and this is kind of his life work, one of his life works, which is this one, calcific aortic valve disease. So as you can see here, this is the 3rd leading cause of adult heart disease. Incidence increases with age, and this is linked to bicuspid aortic valve shown here, and also calcification of the valve. And this exists in approximately 1 or 2% of all the population. And Deepak, a long time ago, found that this 1% of population has Notch1 heterozygosity, Notch1 mutation in one allele of Notch1 gene, as you can see here. So this Notch1 heterozygosity mutation results in congenital heart defects, including valve malformations, and later in their lives, it results in severe valve calcification in adults. So Deepak and his team wanted to utilize iPSO technologies to understand why Notch1 insufficiency resulted in this abnormality. So they published this genetic screening in 2005, and we reported iPS cells in 2006. So it was a very nice timing. So as soon as we published our mouse data, we started a collaboration. So this is what they did. So from patients, they generated iPS cells by those 4 factors. And so they have patient-specific iPS cells. But by using gene editing, they generated isogenic iPS cells. So they corrected Notch1 heterozygosity back to normal allele. So they have isogenic lines with or without Notch gene mutation. And from iPS cells, they differentiated endothelial cells, and they performed omics analysis. So they published the outcome of this disease modeling already 10 years ago. The first author was Christina, who is now one of our PIs at Gladstone Institute. So she found that Notch1 actually suppresses differentiation of osteoblast-like cells from bulb cells. So Notch1, in the presence of Notch1, this differentiation is suppressed. But in Notch1 mutant patients, Notch1 activity is only half. This led to the activation of these transcription factors, SOX7, TCF4, SMAT1, and which resulted in the enhanced differentiation of osteoblast-like cells from bulb cells. This led to increased calcification. So by using this model, so just like ALS, they became able to recapitulate bulb calcification in Petri dish. So naturally, they tested many different candidate drugs, and they identified one chemical, or ERR gamma inhibitor, to be effective. So by adding this ERR alpha inhibitor, these 3 transcription factors, SOX7, TCF4, SMAT1, were suppressed, and osteoblast-like cell differentiation was also suppressed. And they found, at least in mice, it's effective. So in treatment group, the stenosis was significantly weakened. So this is, for the first time, scientists found a small chemical which could treat bulb malformation in adult. So this is going to be the last slide. At the moment, only one available treatment for this calcific aortic valve disease is replacement, valve replacement with this kind of artificial valve. This is very effective, but it's very expensive. So now we may be able to, I'm sorry, not we, but Deepak and his group may be able to change the world by applying this small molecule drug. So their vision, oral daily drug to prevent and treat aortic valve disease. I hope this will happen in the very near future. So in both Kyoto and in San Francisco, we have been working very hard on these two medical applications of iPS cells, regenerative medicine and drug development. We are still on the way. Many applications are now being tested by clinical trials, but it's not the goal. I'm a marathon runner. I feel like we are probably at just at the middle of the marathon course. We still have to run the remaining half. And I know very well that the second half is more difficult, is more severe, but we'll do our best with these many colleagues in Kyoto and in San Francisco. Thank you so much for your attention. Thank you very much.